I'll start by holding my hands up and saying that this presentation is going to be more about questions than it is about answers. So I'm going to try and talk about some of the advances in long-acting delivery which have been achieved across a number of different disease areas, and then try and relate what's been learnt in those areas to where the potential benefits are in the area of TB. So hopefully it'll stimulate a lot of conversation, but very few answers, I'm afraid. So I start off by asking the question of how to define long-acting. This is a very crude analysis here of a crude Google search of some of the terminology which is being used for long-acting. You can see the different terminology down the sides here, ranging from controlled release, sustained release, extended release, time release, prolonged release, and then across the bottom, how they're being applied for different routes of delivery. And the number of Google hits across these different areas. And I think it's fair to say that there's a need for some standardization of the nomenclature here. One of the good examples that I came across, for example, is insulin glargine, which has been termed long-acting for a once-daily administration format. And then the term to trump that terminology was with insulin degludec, which was ultra-LA, because it gives you more confidence about a once-daily administration format. So I think we do need to tighten up around some of the terminology. In infectious disease particularly, and I confess that our thoughts on this originated in HIV, but we're trying to push the terminology for long-acting according to the dosing frequency and do that according to the route of delivery. And we're proposing that for an oral long-acting is anything above 1 week, so 1 week or more; for parenteral delivery, 1 month or more; and for an implant or device, 6 months or more. So if we look at the technologies that have been explored for long-acting delivery across indications, they can be categorized really into those which are administered subcutaneously, such as solutions, or those which are administered intramuscularly, and these encompass suspensions, microparticles, or in situ forming Depo formulations. And then there's a lot of emerging data for device technologies, which I think are emerging quite well, but still have their challenges. There's a number of these which are entering evaluation. These are just two of my favorites. The first one being a microchip type device, which is controlled from outside of the body. And you basically have this chip with these wells containing drug with a biodegradable polymer over the surface. And when you can push a button outside the body, electrodes fire which dissolve the polymer and release the drug into the systemic circulation. And then the one down here, which has been termed an osmotic pump, which doesn't contain any electronics, but the device sucks the fluid in from the external tissue, and as the water gets pulled into the device, the piston pushes the drug out of the device. So a lot of emerging technologies in terms of devices, but in the context of infectious diseases, these devices at the moment at least only able to deliver very small concentrations of drug. So one of the major drivers for long-acting medicines in infection has been non-adherence. And this is summarized really within this PK profile here. If a patient has perfect adherence, then clearly you get good therapeutic coverage, whereas when a patient starts missing doses, then you get into an area where you start to risk emergence of resistance within the pathogen. And so the concept really behind long-acting delivery is that you give this one administration into the muscle, and you get a PK profile which spans the duration of therapy. And it strikes me that in terms of directly observed therapy, there may be benefits there in the tuberculosis area. In one study from Brazil, it was estimated that DOT accounted for up to 75% of the healthcare costs for a TB treatment. And so obviously if you're only administering the drug once a month or once every 3 months, then there's the potential there to cut down on the cost of DOT. Also, in HIV at least, there's quite a strong patient pull towards the long-acting medicines. This is one paper led by our very own Sue Swindells in the front there that looked at patient attitudes towards long-acting injectables. And there really was quite an enthusiasm within HIV patient groups, at least, for the approach of once monthly administration of their antiretroviral drugs. And I should say that this original study has been borne out now in a number of the studies with the newer long-acting antiretroviral drugs, which had integrated patient measures of happiness with the administration. And very recently this week, a big PrEP study which showed positive pull for this type of administration format. So one of the main considerations for long-acting delivery has to be drug potency. I think it's fair to say that probably the single most important factor that drives success of long-acting regimens is the potency of the molecule. I've tried to illustrate that here in this figure. So obviously, the potency dictates the target concentration. And you can see that if you've got a low potency API, then you're very dependent upon the PK of your molecule, because you have to achieve much higher plasma concentrations than you do with a very low potency molecule. So in that regard, if you've got a high potency API, you can be less concerned about the PK of your molecule. Clearly, you prefer to have a long half-life drug, but potency really drives the amount of drug that you need to get on board for a prolonged period of time. One of the critical factors involved in long-acting delivery is the release of the drug from the actual Depo site. This is just a representative PK profile here showing the potential impact of a difference in release rate from the Depo. And this is really where formulation comes in to play. And you can see that with the optimal release rate shown in green here, you equilibrate the release of the drug into the systemic circulation with the clearance of the drug from the systemic circulation, and you achieve this protracted delivery of the drug. Whereas if you've got too high a release rate, you dose dump the drug. And you'd be particularly concerned if you had a Cmax-driven toxicity, for example. But conversely, if you have too slow a release rate, then you'll never get enough of the drug on board to overcome the clearance mechanisms to give you the sustained exposure. So the current long-acting formulations in HIV and schizophrenia and other indications are all based on nanotechnologies. Just to summarize, there are two overarching types of nanotechnology which are being explored in drug delivery. The first is solid drug nanoparticles, where you take a solution of the drug and you precipitate nanoparticles out into a suspension. Or you take big chunks of drug and you just smash them up until the chunks of drug are measurable within the nanoscale. And then the second overarching nanotechnology, which is being explored, is the nanocarrier systems, and these are extremely diverse in composition. They range from lipid-based nanocarriers, polymer-based nanocarriers, inorganics, and many people will refer to many of the biologics as nanomedicines as well, because they're measurable within the nanometer scale. So for the Depo formulations which are emerging for long-acting delivery, what we're talking about is solid drug nanoparticle formulations. So no complex nanocarrier systems, simply nanoparticles which are constructed purely from the drug itself. The most commercially successful way that solid drug nanoparticles have been manufactured is by wet bead milling. It's simply the milling of large particles of drug down to the nanoscale. It's sometimes termed an attrition technique. It's got a number of limitations based on the physiochemical properties of the molecule, but it has resulted in numerous FDA-approved medicines which are routinely used across a number of different indications for either oral delivery or for the long-acting delivery. In Liverpool, we've been using a slightly different approach. I'm not going to go into detail about it, but it's emulsion templated freeze drying approach. And this video here really illustrates the technology. So if you take an insoluble dye, you add it to water, it does what you expect. It just forms a scummy mess on the surface of the water. If you process that dye, so you form solid drug nanoparticles out of that dye, you can already start to see it streaming through into the water. And when the hand comes in to give it a shake, you can see that it's actually, by eye at least, indistinguishable from if it were in solution. But I would underscore that it's not in solution, it's a nanoparticle dispersion of the dye in water. So I just put this slide in to get back to Jerry's point earlier about oral formulations and potential work that needs to be done with rifapentin. But this is some work that we've done with the antiretroviral drug, efavirenz, where we were able to show in vitro in CACO2 cell monolayers that we were able to improve the oral or the transcellular permeation of efavirenz across CACO2 cells, moving into PBPK modeling, showing the potential benefits of that in terms of oral delivery of efavirenz, and then through into in vivo confirmation of that benefit for the oral delivery of efavirenz in preclinical species. And I can say that we've now got human data on this formulation as well, which we're hoping to be presenting at the CROI conference in February. So let's take a look at the existing long-acting Depo agents. They really break down into two separate strategies. We have the prodrug strategies, such as paliperidone palmitate and olanzapine palmoate. This is where the active drug was found to be too water-soluble to be able to make nanoparticles of it. So you can't mill a water-soluble drug. You want basically perfect for the approach is what's commonly and affectionately termed brick dust, because you want to take the brick dust and smash it up into nanoparticles. Obviously, if you take nanoparticles of a water-soluble drug, you're not going to have nanoparticles for very long because they're going to dissolve into a solution. And then other molecules which are deemed to be sufficiently insoluble to just nanomill in their own right, such as the risperidone, rilpivirine, and cabotegravir, these two being the emerging antiretroviral Depo formulations, which are really paving the way in terms of infectious disease for this approach. This is just some summary data here for the cabotegravir, and this really shows the PK benefit of the approach, up to 48 weeks here for cabotegravir, and still detectable levels of the drug within the systemic circulation within these volunteers. If we look at some of the properties of these drugs, I think that can be useful in terms of what we can learn. If you look at the actual characteristics of the Depo formulations here, you've got the dose, shown in this column, and the dosing frequency, which is currently being proposed, so ranging from 2 weeks up to 6 months for medroxyprogesterone. You'll notice that they're all fairly long half-life drugs anyway in their own right, and I think that makes sense in terms of their elimination. The therapeutic plasma concentrations are shown here, so these are those which are usually achieved after oral administration or after Depo administration. The daily oral dose for the oral counterparts of these medications are shown here, and you can see that for their oral counterparts, those doses are pretty low in any case. And then some of the physiochem here, probably most importantly, is the aqueous solubility. These are all low solubility molecules. So I thought it might be useful to look at this on this web type diagram. So these are the same properties: the half-life, target concentration, oral daily dose, log P, and water solubility. And these are the existing long-acting agents which I've just shown you. So if we shade that box, we can think of that as maybe a target area for the properties that we want to get into. If we look at isoniazid, and we superimpose that on the other long-acting depots, you can see that the water solubility is way too high. Target concentrations are probably unachievable by the approach. The log P is low, but the water solubility is really what drives the is driven by that log P. The oral daily dose, perhaps you're not so concerned about that because there's going to be oral bioavailability issues there that might restrict that anyway, and you probably circumvent those. If we look at rifampicin, again, not ideal in terms of the physiochem and the PK properties. Same with pyrazinamide, same with ethambutol. If we move on to some of the newer TB agents, we look at rifabutin, rifapentin, then maybe we stand a better chance here. So you can see that the only thing restricting these really is the daily oral dose, and as I said, that might not be too much of an issue for this. And then delamanid and betaquilin, again, these agents may be more compatible with the approach. So what are the potential benefits? Well, there's a potential overall reduction in dose and cost associated with the regimen. If we look across these existing long-acting medicines and we compare the amount of dose per day from a long-acting regimen compared to the oral regimen, you can see that there's a cost saving across the different drugs, or a dose saving across the different drugs. But what about genetic variability? So this is one study we did with Helen McKilleron, looking at OATP1B1 polymorphisms in relationship to rifampicin pharmacokinetics. I think this does raise some issues when you're thinking through a long-acting approach. I mean, this is an oral PK, but how do we define our target concentrations for LA? We're probably going to have to go back to scratch if we genuinely are successful in getting some of these molecules through to long-acting. Is a 2-hour concentration going to be sufficient as a marker for efficacy? I doubt that. And then there's a potential, of course, that genetics will influence duration of exposure as well as extent of exposure, particularly if the efficacy is C-min driven. So there's clearly some issues that would need to be thought about there. Another thing which is evident from the existing long-acting agents is that, I mean, when I started working on this, I thought you avoid the gastrointestinal tract, you're probably going to tighten up the PK variability. That isn't actually the case when you look at the PK data. So if you look at the AUC variability, it's at least as variable for the long-acting as it is for the oral counterpart, if not more variable. And so that begs the question, you know, what are the mechanisms of drug release from these things? So this cartoon summarizes what we currently know about the Depo formulation. We've got the nanoparticles here which are dissolving into the muscle tissue. We know that the capillaries, which are close to the muscle tissue at the Depo site, the very tight endothelium around these capillaries, probably too small for nanoparticles to directly enter those blood capillaries, but they are dissolving, and so there is a potential to diffuse across into the capillaries. These cells, as well as the muscle cells, are metabolically active, so there could be local metabolism happening there at the Depo site. If we look at the lymph, it's got much bigger intercellular clefts, so the nanoparticles may well be able to enter lymph as intact nanoparticles, as well as by the other processes. We also know that macrophages quickly infiltrate the Depo site, in rodents at least, and that these macrophages are capable of phagocytosing the particulate within the Depo. And shortly after the Depo administration, those macrophages form a granuloma around the Depo, which may serve to restrict the absorption of the drug and slow down the release. So the other thing that we know is that angiogenesis actually happens through the Depo itself. So we've got blood capillaries growing through the Depo after administration. I wanted to comment as well on drug-drug interaction potential. We're all aware of how a drug is absorbed orally. We've got potential for drug interactions in the intestinal barrier in terms of transporters and P450 enzymes, but also we're delivering via the oral route potentially very high concentrations to the liver. So I think there's a valid question there over, A, what the bioavailability is from a Depo site, but B, whether we're going to get those same high concentrations in the liver and in the intestine, which are going to drive induction and mediate drug interactions through this approach. So some key considerations for long-acting Depo. We've got process-specific considerations, and these, I think, have to be considered early. There's some very elegant technologies out there, but they're going to be too costly to implement. So we have to think about cost. We have to think early about scalability, sterility, which we don't have to worry about for oral delivery, and then the compatibility in a technology-specific way with the different drugs. We have to consider drug loading. Certainly in HIV, a major challenge has been the volume of the Depo that needs to be administered, and that is so large. It's 4 mils or two 2-mil Depo sites, so that's uncomfortable for the patient. We need to think about combinations because it's infectious disease, adverse drug reactions, drug-drug interactions. We need to think about the achievable plasma and target concentrations from the approach, the duration of exposure, and again, we've got to think about the target there, not just the plasma, the consistency and reproducibility, the tunability, compatibility with healthcare systems, and then, of course, the patient acceptability of the approach. But somewhere in here, I think, between the drug properties, the formulation properties, and the potential to bring in the evolving device technologies, we're bound to be able to get to a target product profile which is suitable. So opportunities and challenges, if we think about the double-edged sword of long-acting injectables, the potential for dose reduction and cost reduction, avoidance of gastrointestinal adverse events, improved patients' adherence, compatibility with directly observed therapy, and then obviously better protection of health privacy because of the mitigated need for daily dosing. But then some of the challenges on the other side, the interpatient variability, including in time of administration, the large injection volumes and the injection site reactions. DDIs need reinvestigation, I think. The requirement for oral lead-in, which has been heavily debated in the HIV arena, and then the consequences of missed doses for resistance when you've got these very long PK tails. I'll just finish by saying we are funded by the NIH through the long-acting extended release antiretroviral program to really push forward this general area of long-acting delivery. And Sue is heading up the TB working group of LEAP. So we really would welcome feedback on the long-acting approach as this area emerges. And I'll just leave you with some acknowledgements.